Homologous chromosomes are identical chromosomes that can be observed in pairs, in which 50% of this amount is inherited from each parent. Humans have 46 chromosomes, or 23 homologous pairs. Today's lesson is about the non-disjunction of chromosomes. Let's start by the normal disjunction of chromosomes. Well, normal chromosome segregation begins with a diploid cell at the start of meiosis. Let's focus on these homologous chromosomes. Each chromosome consists of two sister chromatids joined at their centromere. During meiosis 1, homologous chromosomes separate, resulting in two cells, each containing one pair of sister chromatids. During meiosis 2, the centromeres split and the two sister chromatids segregate to different cells. The result is 4 haploid cells, each with one sister chromatid, now called a chromosome. In animals, these haploid cells are called gametes. When they fuse with a gamete from the opposite sex, normal diploid cells are produced. Let's find out what happens when non-disjunction happens during the first meiotic division. Let's have a look at these homologous chromosomes. As you can see here, the homologous chromosomes failed to separate during meiosis 1. And as a result, one cell got the two homologous chromosomes, while the other one got no copies of the chromosome. When the cell divides during meiosis 2, the centromeres of both sister chromatids split, and they segregate to each of the two progeny cells. The two cells are unusual because they contain two copies of this chromosome instead of one. In the other two cells produced, there are no copies of this chromosome instead of the usual one. After fusion with a normal haploid gamete, all of the cells produced are abnormal for the chromosome. Half of the cell will have 3 copies of this chromosome instead of the normal 2. This condition is called trisomy. The other half of the cell will have only 1 copy of this chromosome. This condition is called monosomy.